Stuff like caffeic acid has got a lot of interest because it's in coffee. Other ones have got no interest because it's not in anything worth any money. Right on. So, our absorption isn't good. So what's going on, right? So I just told you that they don't work like everybody told you they work. So, simply put, there's not enough polyphenolic compounds being absorbed by the body to reach concentrations within your own cells necessary to inhibit radicals. It just doesn't happen. There's just not enough making it in, right? So the way they assume that it was just meeting up with the radical and quenching it right there on the spot, it's just not going to happen. You need physical interaction of those two molecules, and we've just shown that it's not going to, you just can't get them in there. So something else is going on. So, but we did notice that, however, some of them were getting in the body and were metabolized. That means the body is doing something with them. But what? Especially that they came out in a different form they went in. That's important. Okay, back in 2003, a guy named Howitz came out with a paper. You guys might not know what Nature is, but it is basically the most recognized journal in any field anywhere. Nature. If you get a Nature paper, you've got a full-time job at whatever university you feel like, because they'll hunt you down until they get you. Okay, so anyway, a Nature paper, a huge paper, gave evidence that polyphenols, especially quercetin and another one from red wine called resveratrol, were interacting with a sirtuin pathway. Okay, they did it in yeast. The thing to remember about a lot of these, when I'm going to show some of the organisms they used, we're not that much different than yeast. It's a little hard to think about, but we're really not. We all have mitochondria. We all have the same pieces of instrumentation that Saccharomycetes have. It's a eukaryote, just like us. It actually makes a pretty good model. You're going to be scared. I'll show you some other stuff. We're not that bad. Okay, so they used yeast because it's simple to work with and they're easy to keep in a shelf. You don't have to worry about it. It interacted with a sirtuin pathway, okay? And just like mitochondria, sirtuin is in everything. All organisms have a sirtuin pathway, okay? It's part of the mitochondrial network. It's important for regulating your mitochondria, and it does a couple high-level things within your body. For instance, it controls aging, right? Your mitochondria is your powerhouse. That's where all your energy gets made. You can control the amount of energy made and used by the mitochondria. It has a pretty big incidence on the oxidative potential of your cell. Okay, so we're getting back to oxidation again. I'm not saying that oxidation isn't important for cell viability. It really is. But the way they proposed it working just doesn't make any sense. Okay? So aging is important. It's caused by oxidation. All right? The other thing it does a lot with, and that they've shown, is calorie restriction. Again, this is mitochondrial based, right? So mitochondria cranks out all your energy. So again, calorie restriction would have an effect on your mitochondria. Again, okay? And some generalized stress responses, inflammation, and it can actually turn on your body's own antioxidant enzymes. Let's see if we're going to talk about those right now. Okay. And this middle column is a ratio to control, so how much this enzyme system was activated compared to control. Resveratrol from red wine, the reason why everybody tells you to drink red wine for cardiovascular health, was an exceptionally good activator of sirtuin, or it actually helps to activate sirtuin. I'm not going to get into how it actually interacts with the molecule. I'm sure you guys don't want to know. We can see here on the low end, however, here's our quercetin, our buddy quercetin. Okay, don't be annoyed by the fact that resveratrol is 3 times the activator quercetin is. Quercetin is found in about 10 times the magnitude that resveratrol is, so we're all good.
James, I have a question as
Yep.
you're going here. Resveratrol in red wine.
Yep.
Only made of grapes?
Is that the only place you can find it, you mean?
Yeah.
Oh, no, you can find it a couple of different places. Red wine will do it. Red wine only after it's fermented. You won't find it in the grapes unless you do stuff to them. Okay. An interesting weed we get back in Nova Scotia called Japanese knotweed is full of resveratrol, right up to the gills. Peanuts have resveratrol. They've recently found very small amounts of resveratrol in strawberry fruit, and so the more they look into it, the more and more they start to find it. It's a very odd chemical. It's diverging a little here. It's actually meant to stop fungus. It kills fungus, is what it actually does. Okay, we happen to ingest it and it happens to work and be metabolized within the human body and show all these health benefits, but within the plant it's a little more than fighting off your botrytis blight.
So if we made Haskap wine, red wine.
You guys have already tested for resveratrol in the fruit, and they couldn't find a detectable limit. If you ferment it, it's a different story, but this thing pops up. Sometimes you find it in related species, sometimes you don't. Peanuts and grapes were the first things they found it in. They're not at all related, same molecule. Pine, for instance, where are we at here? Phyceatinol. This is a compound very similar, but actually comes from a gymnosperm. It comes from a pine tree. This metabolate's a very odd one. It sometimes shows up, sometimes doesn't. You can't really tell where it's going to come from. Luckily for us, it's shown some interesting things with getting in the human body. However, the only important part I want you to get about this paper is it has nothing to do with the molecule. Well, it does have to do with the molecule itself, but it doesn't have to do with the specificities. What it has to do is this ring structure. You see this AB ring structure, this ABC ring structure? If the molecule has an A and a B ring in the right area surrounded by some OH groups, it generally does the job. Okay? That's what they were trying to show with all this. And while quercetin isn't a super great activator, there's a lot of it in your fruit.
Isn't Brian Bandy doing resveratrol yeast?
Oh, Brian does absolutely.
No, I think someone on campus is actually researching the yeast part of resveratrol, and he's on James's committee.
Yeah, that's right. Brian is doing that. We have this committee members in nutrition, and one of the stays, besides doing the yeast, is he feeds these to mice, and then he takes their hearts out and sees how long the heart will continue to beat.
Maybe it's something some possible winemakers want to talk to Brian Bandy and see if we can get a resveratrol yeast out of them. I don't know. I don't think he's probably thought of commercialization, or maybe he has.
Cool. So let's get on. Yeah. So Horwitz released this paper in 2003, and the entire field of antioxidants quickly kind of went on its head, because this guy was showing that its interaction with a specific high-level enzyme that's causing your body life expansion and everything. And so a whole bunch of guys who had for a lot of years professed that it was all just like it works in the test tube. None of them got fired, but all of them kind of had to shut up for a while. And so the whole thing kind of switched on its head. Now we do all these studies a little bit different. You notice that Bob said that for our breeding program, we quantify certain classes. Okay, and that's fine for a breeding program because we're really just looking for who the best is, right? You got a lot of germplasm, who's the best, right? But when I do this kind of stuff, I'm not going to show you any of my results today, but when I do this, I actually do the compounds on an individual basis because I'm interested in what individual compounds are showing up, okay? And it's kind of how the science has moved out now. Okay, so some more horrible facts. Quercetin lifespan. This thing is called C. elegans. I'm not going to pronounce that because I can't. Unfortunately, it is even closer to you than the yeast was. Okay? We share so much homology with the genes in this little worm, it would make you a little scared. But anyway, we do. Okay? And so it's an excellent model, just like the yeast. You can keep it on a shelf. It never complains about the housekeeping, never does anything, right? It's just a little worm. This was kind of a weird study because they didn't use the typical entry method that they usually use for this kind of study. Instead of feeding the little worm quercetin, because I guess you can't get it to eat whatever you want, who knows, they decided to just soak the thing in a quercetin solution. Just put them right in a quercetin solution. They used a little molecule that helps your skin absorb compound, and they got a bunch of this inside the worm. Okay? Then they looked at the lifespan increase. It's a pretty interesting paper. And what this paper showed is that that little worm, when soaked in a quercetin solution, survives 15% longer than its counterpart not soaked in a quercetin solution. Okay? They also showed the expression of a couple different genes, most of them involved in antioxidants, your body's own antioxidant system, and they showed that a lot of those were upregulated too. And so they then said that what we think is happening is that this quercetin is turning on a sirtuin pathway, which is turning on antioxidant enzymes. They are keeping the worm alive, and that's why we see lifespan expansion. Okay? And that's what we've been rolling on since then. There's been other studies to back it up, a little bit different way of looking at it. They've shown one directly with resveratrol that actually works better, but it's because resveratrol is a better activator. Anyway, lifespan expansion in this little worm. Okay, so antioxidant enzymes. What the hell am I going on about antioxidant enzymes? You say antioxidants aren't antioxidants, but antioxidant enzymes, blah, 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 right? So the body has its own defenses against radicals. Believe it or not, before human beings were deciding to eat fruit, when we didn't look much different from that worm, we still had a whole bunch of defenses against radicals. Because radicals ain't new. They've been coming at us since the verge of time. Okay? So there's always going to be radicals, all the time. That's just the way it is. Radicals and oxidations are actually necessary for life. Morphogenesis within almost all tissues requires an oxidation stage to finish off your cells. Okay? So you have to deal with this all the time. It's going on in your cells right now. It's going on when you make new cells, when you kill cells, everything. There's always oxidation. It's a natural part of life. Okay? So we developed, or the organisms, all organisms, develop methods to deal with them. For instance, this one here is called superoxide dismutase. Okay? It's got a super high catalytic capacity. It means it happens very fast. Picoseconds. You guys know what pico is? It's like a millionth of a second. That's how long it takes to activate. And it just grabs the reactive oxygen and busts it into pieces, and then the reactive oxygen is gone. Pretty easy, right? Enzyme. Great stuff. Okay? It actually outcompetes the damage caused by reactive oxygen, which, if you guys have any chemistry background, you know that's pretty fast. Okay? So this actually outcompetes the enzyme's damage. We've got a whole host of other ones too called peroxidases and catalases, and as funny as it is, all these enzymes start to get upregulated when this sirtuin is activated. So we're just, instead of having them in our own cells fighting reactive oxygen, it just signals our body that maybe you should produce your own, which are much more effective. Okay? So they're still acting, they're not acting as antioxidants, but they do turn on your body's antioxidant system. Okay? I think that was maybe where I messed up, where I was getting a little jazzed, cherry one. But anyways, antioxidant enzymes, much more effective. They're targeted too, eh? So they don't have to just bump into one another, which is also helpful. Okay, so now we'll get on to the really cool part, and probably, from what I've heard from you guys, is probably where a lot of your marketing might go. I'm not really sure. Anyway, quercetin and exercise. One other really cool thing that came out about quercetin specifically was that it has an interaction with the mitochondria. In fact, it causes mitochondrial biogenesis, all right? When you see the little high school diagrams of a cell, they put one little mitochondria in the corner and say, that's your mitochondria, okay? But that's not how it works. A single cell can have multiple mitochondria, a whole bunch if they want. You can multiply more mitochondria within a cell, and it's not a big deal. Lots of organisms do it. The thing is, the mitochondria, like I said before, is your engine. The more you have, the more a single cell can crank, okay? So imagine, if you will, an athlete who, compared to his rival athlete, has a much bigger mitochondrial count in his muscle cells. That means that his cells can crank out more energy faster than his competitor, okay? Pretty simple. Mitochondrial biogenesis. More mitochondria in the cell. So what does that mean for exercise? Let's find out. So they did this guy here, Davis et al. In 2009, took a bunch of mice, and he fed them quercetin for 7 days, okay? Against a control group, he just fed a regular diet, okay? These mice hadn't been fed quercetin before that. They'd been just grown in a lab. Remember, these are just little white mice grown in a lab, okay? So right before they did the study, they fed the little mice quercetin for a week before, and they saw an increase in the expression of several mitochondrial biogenesis genes. The interesting part is that it wasn't just in the muscle tissue, it was also in the brain tissue. Remember that brain is just a muscle too, it needs mitochondria to crank. It's actually not fundamentally different from the muscles in your actual skeletal muscles, okay? It's just used a little different. Right on. So we saw mitochondrial biogenesis in there. I won't go over the specific gene targets, because it's not going to make any sense anyway. They also did see sirtuin upregulated, though. That was one of the gene markers they looked at. Sirtuin was upregulated. So that's great. The genes were activated. Now, does it mean anything? And yeah, it does. They saw increase in maximum endurance and increases in voluntary activity. This one's a little bit mean. They actually put the little mouse on the treadmill until he passes out from exhaustion, and they just saw which of the two mice could go longer before they passed out. Anyway, our quercetin guy took it.
How long did it last longer?
Like 20 minutes on an hour or something. Like
how long was it till they got exhausted?
A couple hours.
Okay.
You got to kind of leave them alone. The other thing that was much more humanitarian was they looked at voluntary activity of the mouse, and that really has got to do with, you know, your kids are hyper? Yeah. So if you feed them quercetin, they're going to be more hyper. I don't know what else to tell you about that. But anyway, voluntary activity. Basically, when your body has the energy to do stuff, you do stuff. Okay? It's kind of an involuntary activity. When you feed your kids fructose, bypasses your glucose regulator, goes straight into the metabolism. That's why they go all flippo, because they got lots of energy and it's not being regulated. Right? Next time you want to feed your kids some candy. Right on. So this is one mouse study, but we got to keep in mind, right? A mouse is not a human. A little worm is not a human. A yeast certainly isn't a human. None of these things are human, all right? And the problem with a lot of this, especially on the absorption side, is that other organisms have different diets. For instance, mice are typically adapted to eating seeds, where humans are much more omnivorous. Our background diets from when we were still, you know, turning over rocks to get dinner or whatever, our consumption habits were a little bit different. So it's good to vary the type of animal. Sometimes they use dogs, other things. Anyway, when you come across a human study, it's about time you pay attention. Okay?